Namaskar, this is Dr. Rohit and you're watching Scientists Are Academy Towards Scientific World. We have started CUET-PG Life Science Lecture Series and this is our lecture first, Paper Chromatography from the unit first called Techniques. In upcoming days, we are going to upload different lectures on the different units, different concepts for the CUET-PG Life Sciences. Now we are going to understand the paper chromatography. Now here we are going to understand what is paper chromatography, what is the principle, what are the steps involved in the chromatography, how we can perform and how we can pretend the results, how we can conclude the result, that we are going to understand further. So first, what is paper chromatography? Now first time it was introduced by the German scientist Christian Friedrich Schwaben in 1865. Now see, what is the principle? Now principle of separation is mainly partition rather than adsorption. Now paper chromatography is totally based on the partition rather than the adsorption. Substances are distributed between the stationary phase and a mobile phase. Cellulose layers in a filter paper contain moisture which acts as a stationary phase. Now see, this is very important, that what is paper chromatography. So paper chromatography is a type of planar chromatography whereby chromatography procedures are run on a specialized paper. This paper chromatography first time introduced by German scientist Christian Friedrich Schwaben in 1865. And the principle of the paper chromatography is totally based on the partition, not on the adsorption. Okay, and whatever the substances are going to distribute in the paper chromatography are totally based on the stationary phase and the mobile phase. We can see this picture in this image. Down we can see the solvent, right, and little bit up to the solvent we have spotted four samples plus X is the standard, and this is the paper chromatography, right. Now during the incubation, what happens during the incubation, the sample will run towards the top, like towards like upper direction, and once it runs, this results will come out. Organic solvent or buffers are mostly used as a mobile phase. Mobile phase is nothing but the liquid phase or liquid, which is used to move or to separate out the molecules or to move the molecules from one place to the another place. The developing solution travels up the stationary phase carrying the sample with it. Components of the sample will separate readily according to how strongly they adsorb onto stationary phase versus how readily they dissolve in the mobile phase. So overall, the paper chromatography is very important technique, and it is a planar chromatography which is used to separate out the molecules based on the partition, not on the adsorption. In this paper chromatography, mainly two phases are used, stationary phase and the mobile phase. Here the mobile phase is the solvent and stationary phase is the paper chromatography or the paper. Now whatever the sample is there, that sample will move from bottom to top through the mobile phase based on the partition, and through these whatever the molecule components are there, whatever the molecules are there in the mixture, that molecules will separate out, and at the end we will get the separate out the molecules based on the RF value. Now there are different types of paper chromatography. First type is the ascending paper chromatography. Now this technique goes with its name as the solvent moves in an upward direction. Descending paper chromatography, the movement of the flow of solvent due to gravitational pull and the capillary action is downwards, hence the name descending paper chromatography. Third technique is the ascending paper chromatography. Now see, the descending paper chromatography, in this version of paper chromatography, movement of solvent occurs in two directions after a particular point. Initially, the solvent travels upwards on the paper which is folded over the rod. After crossing the road, it continues with its travel downward direction. Fourth type of paper chromatography is the radial or circular paper chromatography. The sample is deposited at the center of the circular filter paper. When the spot is dried, the filter paper is tied horizontally on a petri dish which contains the solvent. Last is the two-dimensional paper chromatography, where the substances which have the same RF values can be resolved with the help of two-dimensional paper chromatography. So these are the 5 types of paper chromatography which are used as per the requirements of the sample. Now how we can proceed it? So let's see how we can perform the paper chromatography in the lab. First thing, the selecting suitable type of development. Now here it is decided based on the complexity of the solvent, paper, mixture, etc. Usually ascending type or radial paper chromatography is used as they are easy to perform. Also, it is easy to handle. The chromatogram obtained is faster and the process is less time consuming. Now here, first thing is we have to select the type of development. Now which type of development we are going to select, that is very important for the movement of samples. So mostly ascending type or radial paper chromatography is used because it is very faster and less time consuming method. Second point, selecting a suitable filter paper. So mostly in the labs we have to select such a filter which is like size of the pore size, and the sample quality should be matched, and on the basis of that we can easily separate out the sample molecules. Third is the prepare the sample. We have to prepare the sample. The sample preparation includes a dissolution of the sample in suitable solvent, in bracket inert, with the sample under analysis used in the making of mobile phase. The sample preparation is very important because whatever the sample we have, that sample as it is we can't put. We have to dissolve the sample with the with the solution, and then only we can use. Spot the sample on the paper. After dissolution, whatever the sample is there, we can put out one spot on the paper at the bottom 1 centimeter, and we can keep it out in the chromatography chamber. Then afterwards the chromatograph will be developed, right? Then paper drying and the compound detection. Once the chromatograph is developed, the paper is dried using an air dryer. Also detecting the solution can be sprayed on the chromatography developed paper and dried to identify the sample chromatography spots. So these are the 6 steps. With the help of these 6 steps, we can perform paper chromatography in the lab. Now this is the picture which depicts the paper chromatography and different pigments and their RF value after performing paper chromatography. Now see, this is the first image which depicts the chromatography chamber, where it shows us paper chromatography or paper. Okay. After incubating the paper chromatography in the chamber, then we will get the sample spots, right? And once we got the sample spots, then we can differentiate, or we can match with the standard. And after matching with standard, we can conclude this first spot is carotene. The RF value is 0.95. Xanthophyll is second one, RF value is 0.75. Chlorophyll A, RF value is 0.65. Chlorophyll B 0.45, and this is the leaf stain. So like this manner, or in this manner, we can separate out the different molecules from the mixture of sample. So this is what the paper chromatography is, where we can use paper for the separation of different types of molecules. Thanks a lot for your attention. In further videos, we are going to discuss with the different techniques for the PG CUET life sciences.